We're going to talk about antimicrobial drugs. The first thing I want to point out is this chapter goes into more detail than what I cover in the PowerPoint. I don't expect you to know every single medication. I do talk about some, and you should know the bigger ones, like, I don't know, penicillin. Like, you need to know what penicillin is and what it does. But the fact is, we're constantly making new drugs, and when you're treating a patient, you're going to have a computer that you're going to look up a particular class of drug because you have to make sure that the patient doesn't have any allergies and there's no medication side effects. So it's more important that you understand how the different drugs target different things when it comes to bacteria, viruses, and eukaryotic pathogens than it is to memorize every single little drug. So try to imagine what our world would be like if we didn't have antimicrobials. Things like pneumonia, staph infections, UTIs. I mean, think about how much life sucked before we actually had antibiotics. You know, doctors could identify what was happening, but there was really not much they could do other than to say, well, lay down and hope it goes away. And unfortunately, misuse as well as microbial evolution is actually threatening these medications. So when we talk about the discovery of antimicrobials, Ulrich, with his creation of salvarsan, was the first documented case of creating a chemical that could actually treat disease. The red dye Prontosil was discovered by Damac in the 30s, and this was used to treat strep infections. It doesn't really have an effect in test tubes, but it was sort of the first sulfa drug. And both of these are chemotherapeutic agents. They are chemicals used to treat disease, but they are considered antimicrobials. Now, Alexander Fleming identified the mold penicillin, and that, well, he identified that it was secreting a substance that was toxic to staph, and he named it penicillin. And he showed it was effective at killing a lot of things, not just staph. He was never able to purify it, so he abandoned the research. About 10 years later, two scientists were able to purify it and test it on a police officer. Unfortunately, they ran out of it, and the patient ended up dying. And World War II actually spurred research and development, and penicillin G was the first naturally produced antimicrobial. It was the first real antibiotic. And there's some cool history behind World War II research. Obviously, you had a lot of war wounds that needed to be treated. But the British scientists that were working on it were always afraid that they would have to be evacuated because of the Blitzkrieg. So every night when they went home, they'd actually rub the mold on the inside of their jackets. So if they ever got arrested or they were ever dragged, you know, to another country to escape, they would still have the mold to continue their research. Shortly after penicillin was discovered, Waxman actually purified streptomycin from soil bacteria. Since then, pharmaceutical companies have really gone nuts testing soils from all around the planet, trying to find antimicrobial substances. We've also learned that by playing with the biochemistry of antimicrobial medications that occur naturally, we can actually create derivatives of them in the laboratory, and that's actually where a lot of the penicillin derivatives come from. Now, when you're picking an antimicrobial, you care about the therapeutic index. So basically, you're looking at the selective toxicity. You're trying to find something that's going to cause more harm to the microbe than to the person you are treating. Now, toxicity is relative, and it's typically expressed as this index. So you want to give the lowest dose that's toxic to the patient, basically divided by the dose used for therapy. So in penicillin, the reason penicillin is so useful is it has a very high index. Remember, penicillin targets cell wall synthesis. It's targeting peptidoglycan. Humans don't have peptidoglycan, so it's not toxic to humans. However, we do have medications that are toxic for us to take internally. Those medications can only be used topically. Now, medications tend to either be bacteriostatic or bactericidal. Bacteriostatic chemicals will inhibit bacterial growth, so they stop growth, but the patient's immune system has to actually kill and eliminate the pathogen. If it's bactericidal, the chemical will actually kill bacteria. These are useful when the host's defenses are compromised or unable to eliminate the pathogens. Sometimes these medications might end up only being bacteriostatic. It really depends on the concentration of the drug and the stage of growth the bacteria is in, if you can actually kill it or not. Now, in terms of spectrum, you have broad spectrum antimicrobials and narrow spectrum antimicrobials. If it is broad spectrum, it affects a wide range of bacteria. This is really important when you're dealing with life-threatening diseases, especially when you have no time to culture or identify what you're dealing with specifically. Unfortunately, it does disrupt the normal microbiota that keeps out pathogens, so it can cause problems. With narrow spectrum antimicrobials, you're affecting a much more narrow range. This does require identification of the pathogen. You might even have to test them for sensitivity to drugs to determine which strain you have. However, this is much less disruptive to the normal microbiota. You can also have effects by combining medication. So if two medications interfere with each other, they are antagonistic. When one medication enhances the other, it is synergistic. Combinations that don't fall into either category are additive. Now, you're going to find that these drugs behave differently depending on where in the body they're going. So only some medications can actually cross the brain-blood barrier. Those are the ones you use for meningitis, right? Because you need to treat a bacterial infection in the brain. Medications that are not stable at low pHs need to be injected to bypass your digestive system. You're going to find that the rate of elimination or the half-life of the drug will dictate how frequently you need to consume it. So basically, you need to keep a certain dose in your body for it to remain effective. But to maintain that dose, that's why you have to take it every 6 hours, every 12 hours, whatever that dosage is. If you have kidney or liver damage, the dosage you take and how frequently might be different so that you're not poisoning yourself. You can have adverse effects to antimicrobials. This includes allergic reactions and toxic effects. So unfortunately, you can have suppression of the normal microbiota, and this allows dysbiosis. So Clostridium can actually become opportunistic when your normal microbiota is suppressed. And it is important to remember that these medications have saved countless lives when they're properly used. Now, unfortunately, there is some resistance to antimicrobials. Now, some bacteria just have innate intrinsic resistance. Mycoplasma does not have a cell wall. Well, if you don't have a cell wall, penicillin's not going to do a darn thing because there's no peptidoglycan in a cell wall. If you look at the outer membrane of gram-negatives, a lot of them can block certain medications. You can also have bacteria acquiring resistance. This happens through spontaneous mutations and horizontal gene transfer. Let's pause here.